Joe 508, hi, how are you? As promised, I'm coming back to explain a little bit more this concept of how um, our lives and how we live them can alter these switches that exist in our DNA. Because I think most of us, we think about our DNA, um, we're given this DNA with for our lives, and it's the same exact DNA all throughout every cell in our body. Kind of the way to look at it is the DNA is like this giant cookbook, okay? And it has 100 different chapters, okay? And what makes a bone cell, a skin cell, um, a brain cell, has to do with opening up that cookbook and which recipes are you going to read um, to make the new proteins, okay? Um, and those recipes can be from any chapter. Probably instead of the number of chapters said, maybe like 23 chapters, okay? Like the number of chromosomes, okay? Like 23 in me, okay? And that's how it works, okay? And we'll go through, we'll see how developmentally which chapters and which recipes are read change developmentally, and it helps determine that this cell that was a stem cell is going to become an immune system cell, a red blood cell, a skin cell, a muscle cell, things like that, okay? And then the performance of these cells is then altered by a lot of things that we're exposed to in the environment, okay? It can be diet, okay? It can be environmental insults, chemicals that are toxic, and it can be our own internal environment and the levels of hormones that are fluctuating in our bloodstream. And the real focus of this class is the one internal environment hormone that we really try and control through things like meditation and wellness in general, exercise, and that is the stress hormone that's called cortisol. Okay, so that's kind of the background behind that. All right, let's now go back into the actual course, okay, and try and get some clarity. Now, again, I want to say to you, just read this and have as much pleasure in reading it as you can. The quizzes are just a way of checking to make sure that you're reading it. Super performance on the quizzes is not as big a deal as you might think, okay? The bigger assignments are the midterms. They are completely open book, the two of them. And then, the two writing assignments. Again, and every step of the way, we understand that you guys are not biology majors. And if you would just lower your stress, okay, reduce your cortisol, and just try and take it in as best you can, you're going to learn a lot, okay? And that's the approach that you need to take, okay? All righty. So we're going to go back over here to the readings, and we're going to go to the discussion, okay? Now, in terms of the discussion, let's go to this real quickly, okay? All right. So we're here on the second discussion, okay? And so you first got exposed to the behavioral aspects of stress up here in the Sapolsky videos and the psychological aspects. And I'm going to give a little clarity on that as well. And we're going to go over this over and over again, how the brain works and how we sit there and think about things and create this environment of stress that really impacts all aspects of our body's physiology. We're going to do it again, so don't worry about it, okay? This gets into a really unique concept that a lot of people don't think about, and that is this kind of stew that we ruminate in, okay, when we're really stressed out, okay, can really affect the performance of every individual cell by opening up that cookbook, by going into the DNA and saying, we are going to make this protein and this protein and this protein, and we're not going to make this one, this one, and this one. And that's what these little switches called methylation switches do, okay? It's not the only one out there. There's another one called acetylation. We're not going to get into that in this class. But these little switches, you don't mutate the DNA, okay? The DNA stays the same, but it just says which chapter are we going to, and within that chapter, which recipes are we going to be cooking to make new proteins. And sometimes you can make too many of these proteins and can cause some serious health conditions. Sometimes you can make too little of other ones that causes serious health conditions, okay? And that's the whole essence of this epigenetics section, okay? So I'm gonna go through the Utah site. It's an amazing site. You just, again, same deal. Don't stress out, okay? Just take it in, my friends, okay? So we ask you, in the prompt, okay, kind of discuss the sequence of events that goes from a psychological stress to changes in DNA function, all right? So psychological stress, okay, what are we talking about? We perceive something in our internal environment, okay? And we're gonna learn all about this fear center in our brain called the amygdala, and it sends these messages rifling down to the hypothalamus. And hypothalamus interacts with our neck down system via the hypothalamic pituitary adrenal axis, okay? And that ultimately from the adrenal gland releases this hormone called cortisol. And cortisol goes into cells and changes the function of DNA. Key element right there, okay? So we're gonna go through this, okay? It binds to a receptor, it goes in there, it changes which chapters are gonna be read, okay? There's a thing called the transcription factor. And we're gonna transcribe new recipes to make new proteins. But it also goes in there and starts flipping these methylation switches, okay? All right, so that's key. Now, we're making sweeping generalizations, okay, guys? There are times when turning on that cortisol receptor can be a good thing, okay? So we learned about how maternally, okay, when you are an excellent parent, okay, and you are there for your child, you're a rat, and you lick your little rat pup, okay, that you can increase these cortisol receptors. They're called glucocorticoid receptors, because that's what they're called in rats. In humans, we call it cortisol. You increase the number of those in the hippocampus, and it allows the hippocampus to shut off that stressful response that exists in your brain. That's the key concept there, all right? All right, so we want you to talk about, I'm going to show you step by step how this works, okay? And how it gets into these molecular sections, switches, sorry, okay? How it binds to the proteins, okay? Methylation tags, binding to the proteins. And then you come up with some ideas how genes get turned on and turned off that cause acceleration of aging, like inflammation, okay? Big one right there, okay? Alrighty, so you can be stressed out and create an increase in inflammatory signals, okay? And cause inflammatory diseases like autoimmune disease, okay? Like atherosclerosis, and then that leads to heart disease, stroke, kidney disease, things like that. So that's kind of like where we want you to go, all right? All right, so that's the concept of the discussion, all right? So now I'm gonna come over here to, again, the readings, okay? All right, and so your background is this right here. It's the epigenetics section. So we get in here, you watch that video. It's pretty straightforward, okay? You click on this, okay? All right, so this takes you to this Learn Genetics chapter. You watch this video right here, okay? So it comes up and up comes the video. All right, gonna go backward on my, my browser here. It's kind of fun to look at the gene control section, okay? All right, so for me, you have to be able, not all computers have Flash. If you don't have Flash, don't freak out. All right, so you can go like this. I'm gonna allow Flash to play. Okay, now we're gonna look and see what happens here, okay? So when there's all kinds of methylation like this, all right, we see that there's kind of no reading of the DNA. Now this fluctuates, opens and closes, okay? I can only read the DNA if I open it up. And the proof of this, of course, is looking down here, okay? So we can watch how many recipes fly off the DNA when I open it up. Boom, 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 boom. See all the new recipes, okay? Those recipes were from my cookbook. That is this thing right here, the DNA, okay? And when we go like this, More recipes, the more proteins we make. We use this thing called a green fluorescent protein that we can just put on the back end of the recipe. So every time I make a new protein, I make a green fluorescent protein as well. And that's how science kind of is able to watch which proteins are made and when. That's what that's all about, okay? All right, so that's my camera. I'm gonna turn it off right here, all right? All right, so that's what that one's all about, okay? Then we go into how the epigenome works. So like I said, we start out with these stem cells, and if ultimately they're going to become lots of different cells. In this particular example, one type of neuron, or maybe it became a muscle cell or a kidney cell, and all along the way, remember, every cell has the exact same cookbook, the exact same 23 chromosomes, okay? However, as we go along the way, we're changing these epigenomic tags, these methylation tags, saying yes, no, yes, no, yes, no. And next thing you know, you have a nerve cell or you have a kidney cell or whatever it is, okay? And so this talks about it right here, how we go through this development of gene expression, okay? This right here talks about development, and then it talks about how even later on, things that we do in life will cause these switches to change, okay? So what's your diet like? Do you exercise? This changes gene expression through these epigenomic tags, okay? And that's what that's all about, okay? And this explains it right here a little more clearly, okay? How do we go about doing this? Well, one way to do this when we talk about stress, this could be my stress hormone, right? So this could be cortisol. It binds to the cortisol receptor. We'll see later on, they also call that a glucocorticoid receptor. And it's going to march through the cell and it's going to regulate what aspects of the DNA are going to be read to make new proteins. All said differently, it's going to go down to our cookbook. It's going to determine which chapters opened, and not only within that chapter, which recipes are going to be read. All right, so that's how that works. And this shows you right here what it's doing, okay? This whole concept of we're going to either turn genes on, we're going to either read that recipe and make new proteins, or turn them off. And it deals a lot with these tags, okay? If the epigenome has methyl groups on it, you can't open up the DNA, you can't get to the recipe. The cookbook is closed. All right, awesome. So that's what that's all about right here, okay? We then get into inheritance, okay? And it talks about the importance of things that happen early in life. So we did our nurturing behavior in rats, all right? And we'll go through this. We talk about how that affects methylation, and it also affects stress responses, okay? Like life isn't hard enough on women already, and now here it is. Everything you do during your pregnancy is not only affecting your health, affecting your fetus's health, but it's affecting your grandchildren's health by flipping these epigenetic tags. And potentially, research is showing that your grandchildren, okay, when they develop and pass it on to the next generation, some of these tags make their way through, okay? So that has pretty strong implications for evolution, all righty? Cool, all right, that's what that's all about, okay? This is really important, really interesting, I thought. Again, read this at your leisure. Do not freak out on this stuff. I just want you to enjoy it. But the fact, you sit there and you think, God, some of the traits of my child David looks just like me. Okay, he has my kind of hair trigger temper, and I have one of those sometimes because that's kind of what I saw with my dad. My wife has been able to epigenetically modify me, so now that I'm through cognitive behavioral therapy, I'm really good now. I promise. Okay, but he kind of has that a little bit. I have ADHD and have trouble reading sometimes. I really grasp figures, but sometimes, to this day, maintaining my focus. My wife, Julia, who you know is your co-instructor, is amazing with reading, and David is amazing with there. So he's gotten both of these traits, and it's interesting how it continues throughout the child's development based on certain specific imprinted genes, and that's what that means. So these traits, okay, and that's what this is all about, okay? And it kind of just goes through it. Read this at your leisure, okay? All right. It does talk about things like this, okay? Environmental signals, diet, exposure to toxins, and these can then be passed on, okay? It's important to realize that. Boom. Okay, so that's the imprinting, okay? We look at identical twins, and yet we look at the expression of methylation in them, and it varies. So it shows you the effect, the importance of environment. So please, on your leisure, watch this video right here, okay? And read up about nurture and nature and the differences that happen, okay? So this shows you the close identity of the genome, okay, and identical twins at 3 years of age, but that it drifts apart with aging because of life experiences, okay, and that has to do with the epigenetic tags, and it goes through a lot of different aspects. We're comparing identical twins here to fraternal twins, and we see that height is highly genetic, okay, all right, but we look at things that come down here, and we start to see this genetic drift where there's not such a big percentage, even for identical twins. You see diabetes, breast cancer, Crohn's disease. There's not that much correlation here between the twins because of these, even though they have the identical cookbook, but the recipes that are being read, the epigenetics tags that are being placed on based on their separate life experiences affect these things that happen later in life, okay? That's the concept there, all right? You see early disabilities, okay? Reading disability, autism, there's a much higher, they call it concordance or agreement between the identical twins because that's not such an epigenetic. It's more a true mutation in the DNA, okay? And that's different than epigenetics. And that's what that's all about. We saw how we licked our rats. It was awesome. Okay, it's loading because I did my my flash. Some of you don't have flash, so it's not going to work. Okay, don't freak out. Okay, we look at different things right here. Okay, certainly as we look through this, we know that it affects our ability to nurturing affects our ability to respond to stress. Okay, and when we come down here and we look at that, okay, we can also take a relaxed, high nurtured rat, inject it with methionine, okay, and make it more anxious, okay. This varies, okay. However, this methionine story is not so simple because lots of methionine in the diet seems to protect us, okay. And I did get a question on that, and I do understand the confusion with this, okay? So it's not a clear-cut story. And this is where we get down to the glucocorticoid response right here, okay? So in keeping with this theme, so we injected this thing with methionine. Let's take a look at nutrition, okay? All right, so this is the nutrition thing, and I have something else to still make this easy. So we talk about B vitamins, the folic acid cycle, and now when we have food that's high in folic acid, it drives our source of protein to make more methylation, to turn off unnecessary genes, okay? And that's kind of what that's all about. And we see right here that we can feed a mouse, okay, that has this issue where it doesn't have much methylation. It's yellow, it's obese. We give that mouse lots of methyl sourced foods. You see this methyl rich diet, and we see that the pups were brown and stayed healthy for life, okay? So this is different than that injection story. I'm sorry it's not clear cut, okay? All right. Okay, and we can kind of go through all the different aspects of nutrition here in depth, okay? Awesome. I'm not gonna go into that. So we did the nutrition. and then we can talk about brains. And this is, again, this is a great video. Please watch it if you can. Sorry, I thought I turned that off. Okay, I did now. All right. And it just talks about things that are way beyond the scope of what you need to know. So just read it, listen to it, and take it in. Okay, does that sound awesome? Sounds awesome to me. Okay, so that's the basics on that. All right, so then I'm going to get out of here. I'm going to go back here. Okay, we're going to go into this paper. So I'm going to click on the paper right there. Okay, and I'm going to move my head over here, and we look at the essence of this paper. All right, there are two things. Okay, we're looking at stress, epigenetics, and aging, and that's what the goal of this thing is. Okay, so in this review article, read it. Do not get lost in the facts. Okay, we're going to build upon existing evidence. Okay, where life stresses via the hypothalamic, pituitary, adrenal axis, okay, and the glucocorticoid signaling system, okay, may alter the epigenetic landscape across the lifespan and influence gene regulation, okay, all right, and this functions in ways that are conducive to development of age-related diseases, okay. So this is different than that scenario where we, injected the rat pups with the methyl group. This is totally different. I want you to understand that. Okay. All right. So as we go down here, you can go and just ease into this. Do not worry about it. Okay. Stressing the epigenome, focus on glucocorticoid signaling. So this right here is just a review. Of what goes on. So you read through this, and it talks about the hypothalamus, the pituitary, the adrenal axis, the HPA axis. Okay, and ultimately it describes how the hypothalamus releases ACTH. Okay, the pituitary releases ACTH. Sorry about that. It runs down to the adrenal cortex, and that's what the ACT means. Okay, so this is the adrenal corticotropic, so it's going to the adrenal gland, and it causes a release of cortisol from the adrenal gland that sits on top of the kidneys. Okay, we see here that there is evidence of linking this to methylation. Oh, this stuff is so deep and so beyond your needs or realm. Just read through it and pull out the essence of it, okay? We see that DNA methylation and aging. So with aging, the level of methylation goes down and creates bad things. That's kind of our view on this in this article, okay? All righty. All right, and we look at linking stress in life to this, okay? Again, so just read through this real simply. Don't get too engrossed in the details, okay? So stress, the cortisol binds to the receptor. It gets internalized. This thing goes down to your DNA. It says, let's read certain types of things, okay? It says in order to read these things, we're going to get rid of the methyl groups, okay? And over here, it's gonna inhibit methyl functioning, okay? That's the essence of this, okay? So it opens up the genes and causes the reading of way too many bad things. Okay, all right. That's what that's all about. Now I want to go back to this real quickly, okay? When we look at nutrition in the genome, I did want to go back over here, and if we want improved methylation, what kind of food can we do? So what I did is I went over here and I just clicked on this, okay? All right, and it shows you all these great foods: sesame seeds, Brazil nuts, fish, pepper, spinach, leafy vegetables. Everything in here looks wonderful in terms of donating methyl groups. This is something else right here that we're not going to get into. This is the acetyl group, okay? So we all know those are good foods, and this is a mechanism how they help, okay? All right, and that was attained by going to this nutrition segment right here. Okay, just wanted to go back to that. I should have done that earlier. I apologize. Okay. So I'm going to post these two figures along with my video so you can just kind of study them. All right, here I was trying to get some clarity. We're going to get into the brain mechanism in much greater detail, and it's going to be easy for you. Easy peasy, I promise. Okay. All right, so I'm going over here to a PowerPoint that I've created. All right. Um, so when we are stressed out, okay, this right here is our anger center. It's also our fear center. It's called the amygdala. Okay, it sends a signal down to the hypothalamus. And this is the hypothalamic pituitary adrenal axis. Okay, so it activates the hypothalamus tells the pituitary, sends a message to the adrenal gland releases cortisol. And cortisol has all these changes that are good if you're being chased by a lion, good if you're highly stressed at a moment, but not good to be, as Robert Sapolsky said, to be tapped into chronically day in and day out because you're worried about paying your mortgage, or you're upset because your girlfriend or your wife or your boyfriend or your husband or whatever it is has been treating you poorly. Okay, that's not what it's designed for, but that's what we use it for. So we secrete all this cortisol, and it has all these, as we're going to learn, undesirable effects like diabetes, heart disease, immune suppression. Not a good thing. Okay, all right. So what we do to keep this in check is once the stress is over, like the lion has stopped chasing us, okay, then we turn around and we feed back and we turn the system off. Negative feedback. And over here, I put this in right here, and this is from our learned genetics side, okay, that if we have more of these receptors in this one tissue, in this one part of the brain, okay, the hippocampus, the cortisol feeds back. It's more effective for the hippocampus to shut off the stress. That's the key concept right there. Okay? All right, I've put the links right here, and you're more than welcome to go there. Okay, so there, awesome. Okay, so I'm going to get out of this. All right, and I'm back to our website. Okay, all right, so that was part one. Okay, so then I'm going to go back to this. Sorry, I should have stayed in there. I'm just going to bring it up here since I don't want to have to hit my escape sign. And this makes this figure from Learn Genetics maybe a little easier. So pregnant women all know they're supposed to take folic acid so that they have the right level of epigenetic switches in their DNA. This is what it's all about, okay? All right, we have our B vitamins you see right here, okay? So, and we have certain levels, certain proteins like nuts we saw before, like certain fish that are high in methionine, okay? So what happens is when we have folic acid, it's going to drive the system to go this way along with our B vitamins, okay, so that we make lots of methionine. We combine that right here, and the methionine makes the SAMe. We see the SAMe right here, and the SAMe is on this assembly line to produce these methyl groups, okay? All right, once it comes down here, it has to be recycled and redone again, recycled and done again, okay? When you're down here, you don't have the methyl group, okay? So we're taking B vitamins to drive this back up, and we eat certain proteins to make sure we have an adequate supply. I have circled this right here because anybody that has had vascular disease, heart disease, okay, you've had a heart attack, stroke, homocysteine is a bad thing, okay? And that's why taking our B vitamins takes out the homocysteine. We convert homocysteine into methionine with our B vitamin, which is wonderful, okay? And at the same time, we provide more methyl groups so that this happens right here. This is important for stability of our DNA so that you don't get mutations right in here. And it's also important in terms of which recipes we're going to turn on and off with our methyl groups. Okay. All right. So that's what those two figures are. I looked at this one, okay, right here, and we looked at this one, and I'm going to put those onto the website as well. Okay. Now we're going to go back to our website, okay? So I hope that has helped a little bit, okay? More than anything, I am here to put your mind at ease because I want you guys to do well, okay? And I want you guys to understand that we do not have this expectation of a biologist perception, but isn't this wonderful that you've now been exposed to it and you can bring this up at parties, okay? And with that, we are going to say goodbye.